Biological computation is a novel and increasingly popular technique within synthetic biology for synthetic gene networks and biotechnological engineering. Biological computation is defined as engineering biological systems that are capable of converting inputs to outputs, similar to that of a circuit board but within cells. Logic within synthetic gene networks is often employed at a transcriptional level within the cell, using transcription factors to activate or repress genes of interest. Through the use of inducible transcription factors, transcriptional logic gating within gene networks are able to perform Boolean logic, creating genetic equivalents of and, or, and not functions among many others. Scientists are able to engineer genetic programs that give a desired phenotypic response within host cells through controlling the cell's input. Currently, there is a limited range of inducible transcription factors that scientists are able to use in the generation of these transcriptional logic gates. Thus, intense efforts have been made to expand the genetic toolkit and make more transcriptional logic gates for synthetic biology. One of the recent novel methods involved taking advantage of the different ligand and DNA binding sites of a family of transcriptional repressors. A study published in 2014 by Schiss et al. Takes advantage of one commonly used inducible transcriptional repressor, LAC-I. LAC-I binds the LAC promoter and prevents transcription of downstream genes by blocking RNA polymerase binding. In the presence of its ligand, alactose, or its non-hydrolyzable equivalent, IPTG, LAC-I loses its affinity for the promoter. The presence of the ligand de-represses the promoter region, allows RNA polymerase to bind, and transcription to proceed. LAC-I, which regulates the LAC-operon, belongs to a wider family of transcriptional repressors that regulate sugar metabolism. Each member of this family has their own sugar ligand and respond in the same way that LAC-I responds to IPTG. Schiss et al. Showed that chimeric transcription factors can be modularly constructed by replacing the ligand binding site of LAC-I with those of the homologous family members. This created a library of chimeric transcription factors that responded to different sugars while regulating the LAC promoter. In the study, they were able to make modular logic gates using the chimeras, creating 2, 3, and 4 input AND gates that were much simpler than the conventional existing technologies. Each chimeric transcription factor only responds to its own ligand and are orthogonal to the LAC promoter, which enable multiple types of AND gates to be produced. As shown in the figure, through combining 2 chimeric transcriptional repressors, a 2-input AND gate can be produced. For most combinations, only when both ligands are present can you see a high increase in GFP production of between 14 and 27-fold, thus working as a functioning AND gate. Similarly, through combining 3 transcriptional repressors to the LAC promoter, a 3-input AND gate can be produced. In the study, they combined the original LAC-I repressor and 2 chimeric repressors with fructose and ribose ligand binding sites, producing a functioning 3-input AND gate. Only when all 3 ligands are added, shown in the last column, the promoter is de-repressed and GFP is expressed, shown by an increase in fluorescence. The study went even further and were able to create a functioning 4-input AND gate through combining LAC-I with 3 other chimeric repressors. Again, high fluorescence of GFP is seen only when the 4 appropriate ligands are added. In addition to altering the ligand binding site, the DNA binding domain can also be mutated to create an alternative DNA binding site. 3 existing residues within the DNA binding domain were mutated into T, A, and N residues, called the TAN mutation, resulting in a differing DVD and promoter region. The study showed that these chimeric repressors with the different DNA binding site were also only responsive to its own ligand and were orthogonal to the TAN promoter. Thus, they were able to create functioning 2-input AND gate with these new transcriptional repressors. Having the option for alternative DNA binding sites other than the LAC promoter opens up doors for co-expression of repressors and layering of AND gates to create larger and more complex genetic circuits and logic gates. These novel tools increase the simplicity of genetic circuits and provide a wider range of inducible transcription factors with different ligands and binding sites for use in synthetic gene networks. As the modular construction in this paper was based on the LAC-I repressor, all the chimeric transcription factors are orthogonal to the LAC-operon and can be used in combination to create much simpler multi-input AND gates. However, modular construction of transcription factors is not only limited to ligand binding. The DNA binding domain can be mutated to change the DNA recognition and can be altered to recognize a different promoter. Overall, this increases the scope of implementing logic within synthetic gene networks by increasing available ligands in DNA binding regions.